Hello students, today I am going to talk about Agrobacterium tumefaciens mediated transformation, particularly the TDNA transfer mechanism. So there are two major steps involved in Agrobacterium transformation. One is what are the things happened in Agrobacterium and what are the things happened in plants. First we look into the whatever happened in the Agrobacterium. So there are three major steps involved in Agrobacterium. First step is perception of wound signal to initiate virgin induction. So the phenolics and other plant derived compounds that were released from the wound site of plant, where the plant damage that will release some compounds. So those are mostly phenolics. These phenolics have a pH around 5 to 5.8. This will attract by the agrobacterium. Some bacterial genes also produce different polysaccharide that aid in the firm attachment of agrobacteria onto the plant surface. Upon attachment to the plant surface, the chromosomal virulence genes from agrobacterium, that is chromosomal virulence A, chromosomal virulence B, and polysaccharide A genes that lead to the production of different polysaccharide even for the attachment to be making stronger. The cellulose microfibrils are synthesized by Agrobacterium tumefaciens at the wounded site. This was confirmed by mutation studies. So apart from bacterial proteins, some plant protein also involved for this attachment. For example, vitronectin or vitronectin like family protein 1 that was found to be involved in the initial attachment colony forming process, and many glycoproteins also play a role in attachment. Once the bacteria attaches to the host, that is a plant, the signals were perceived by the transmembrane receptor Vir-A. The autophosphorylation of Vir-A activates Vir-G, a two-component signaling system. Vir-A and Vir-G act as a two-component signaling system. The proteins chromosomal virulence G and chromosomal virulence I are essential to activate the basal expression of Vir-G expression. The phosphorylated Vir-G activates the downstream Virbox genes. In the second step, the processing of tDNA from TA plasmid. So agrobacterium have TA plasmid that is flanked by that contains ORI and marker and tDNA. tDNA is flanked by right border and left border. The tDNA was excised from the TA plasmid by Vir-D1 helicase that has a helicase activity and Vir-D2 that has a nuclease activity. That nuclease is single-stranded nuclease activity. The tDNA was sliced between third and fourth base of identical repeats in the right border and the same time in the left border. The 5' end of the covalently attached to the Vir-D2 and the tDNA complex. Vir-D2 is covalently attached to the tDNA complex. The Vir-D2 comprises 3' nuclear localization signal which direct the tDNA to transfer from cytoplasm to the nucleus. Vir-C1 binds to the overdrive sequence near right border. And the third step is export of the tDNA complex to the host by type 4 secretion system. The tDNA complex along with the Vir-D2 direct the transport to the plant cell through type 4 secretion system. The type 4 comprises of Vir-B and Vir-D4 proteins. The recent identification of Vir-D2 binding protein, that is VBP, form a dimer with the cytoplasm of Agrobacterium and recruit the Vir-D2 tDNA complex to the T4 secretion system through the interaction of Vir-D4 proteins. So up to this happened in the agrobacterium, and the next steps involved in plant. After it is entered into the plant, what is happening in the plants, we will look into another 3 steps. So the first step is export into the plant cell. The type 4 system allows the Vir proteins, that is Vir-D5, E2 and E3 and Vir-F from agrobacterium to the host cell. The type 4 secretion system has some functional proteins such as VIRB complex and VIRD2. These functional proteins act as a substrate, receptor, inner membrane translocation, outer membrane complex, and pilus formation. VRE2, a single strand binding protein covalently attached to the tDNA complex and prevent from the host nuclease activity. The VIRB complex and VIRD4 is essential for the transport of tDNA complex into the host. And the next major step is the cytoplasmic trafficking and nuclear targeting of T complex. The tDNA complex interacts with the additional viral protein that are transferred from bacteria to the host cell. VRE2 interacting protein, that is VIP1, interact with the VRE2, thereby preventing the activation of plant defense gene. Before integration into the host genome, the effector proteins were essential for the nuclear import and also in the export to the host nucleus. The tDNA complex import in alpha mediated, that is through the nuclear pore with the help of cellular proteins. Initially, we will need to NLS sequence interact with the import in alpha and import into the cell nucleus. And the last step is tDNA integration into the host genome. The tDNA integrated at a random site, either transcriptional active or hypomethylated regions of the host genome. That's why it is called illegitimate recombination. Some important modifications occur in the chromatin and histones upon tDNA integration. In nucleus, the protein components should be disassembled by the ubiquitin proteasome system. The disassembled and VRE2 degradation process was assisted by VRF. Sometimes VRE3 used as a substrate for VIP1 that facilitate the tDNA integration. The tDNA alone integrated into the plant genome by non-homologous enjoining method, or else it is called illegitimate recombination or random integration. So the overall steps were recently Huang et al. Reported. This is updated mechanism. So once from the bacteria recognize the signal. So once recognize the signal, the phenolic signal, the VRA activate VRG, then VRG is phosphorylated, then it will activate the further VIR genes, activate the VIR genes in the TA plasmid. So the tDNA is processed within the Agrobacterium and it is generated single-stranded tDNA along with the VIRD2 nicking. So VIRD2 has a nuclease activity and it has a NLS signal. So when it is attached to the plant surface, some plant protein as well as some agrobacterium proteins also involved for the firm attachment. Once it is entered into the plant cytoplasm, the VRE2 that also bind with the single-stranded tDNA to prevent the host cell damage. Once now it is called T complex. This T complex is interacted with the plant VRE2 interacting protein. So that has an NLS sequence for nucleus. Then it will export into the nucleus by importing alpha protein. Then it is export inside the nucleus. Then the VIR proteins are removed, only the tDNA alone it is integrated into the host genome. So this is the mechanism, proposed mechanism, but still some controversies are there. There is not a single tDNA along with some double-stranded tDNA also integrated, not that also reported. So till now this is the accepted mechanism for tDNA integration. Thank you.